Hello everyone and welcome to my video, and this video is basically going to be talking about HIV and other viruses versus humanity and our battle of innovation and the fight for new medicines. So before I get into existing treatments or the sort of a solution that I'm trying to solve in general of educating people and innovations of medicine, I'm going to first give a quick, I'm going to give a detailed explanation on what HIV is. So first of all, what is HIV? So HIV stands for basically, it stands for human immunodeficiency virus, and it's a virus that weakens your immune system from fighting off certain infections or illnesses. So this is a problem. So today, actually around 1.1 million people living in the U.S. have been diagnosed with HIV. This actually doesn't account for the many that are probably living around asymptomatic and not knowing that the virus exists inside of them until it's too late. Until it's too late. So HIV is itself is a virus that affects a certain type of T cell immune system known as the CD4 cell, and it's an RNA virus, and so its genetic material is RNA. And there are some differences between DNA and RNA viruses, such as RNA has an RNA genetic material, and it tends to mutate a lot more without a certain proofreader in one of its enzymes. So specifically, it's classified as something called a lentivirus, and lentivirus is basically a virus that can cause diseases very slowly. So now we're going to talk about some of the history of this virus, how it came to be. As we might know, a lot of viruses originate from certain animals. For example, we recently found out that the coronavirus came from bats. That were then somehow the virus was brought into our system from them. So in this case, although these are all hypotheses, people believe that HIV originated from some type of either chimpanzee or something called the rhesus monkey. So the first possibility that people talk about would be that a specific subtype chimpanzee that lives in Africa would carry this virus that is similar to HIV, and then somehow this virus is brought from this chimpanzee to this human, and this human would get this HIV and would not be resistant to it. The next possibility is having to do so the second possibility. So as we know, there are four different blood types. So I'm going to explain this possibility. So we have blood type A, AB, O, and B. And we also know that there are positive blood types and negative blood types. So there's Rh positive and Rh negative. So let's give a bit of I'm going to be theoretical here. I'm going to give an example. So let's say two people have a baby. Female identifying, male identifying with those parts of female and male. So let's say this female is Rh negative and the male is Rh positive. So they have their first baby, and if this first baby, let's say this first baby is Rh positive, it will pass certain antibodies into the mother's blood against this Rh negative because the blood types are different. So it will pass these antibodies into the mother's blood. So this isn't going to harm the mother, and the antibodies those antibodies themselves can break up red blood cells, and this is not going to harm the mother. But, so let's say you have another baby, and this next baby is also Rh positive. So these antibodies are here, and they could actually clot this they could actually clot this baby's blood, and this can severely damage the baby, and it will be very dangerous. It causes something called hydrops, and the baby can become swollen, anemic, and there could be many other problems that occur. And if this baby isn't treated with some type of blood transfusion or something similar to that, the baby will die. So this is something that happened to many, many, many people around the world before there was a certain treatment created to get past this. And so the treatment that they created is known as ROGAM. So basically, it gets this substance that blocks the antibody, and they found this treatment from a monkey known as the rhesus monkey. So they take this substance from this rhesus monkey and bring it into the baby and block these antibodies from harming these red blood cells and breaking them up. So what we didn't know is that this monkey, this rhesus monkey, might have been the cause of bringing HIV into our human lifestyle. So not only would this monkey bring this antibody blocker, it would also bring in possible HIV. So those are some possibilities of how HIV came to be, how it came to humanity. So now that we have a sort of now that we have a bit of a sense of what HIV is and some of the history, how it came to be into humanity, I'm going to go into a bit about the stages of HIV, of infecting a human. So the first stage of HIV would be the infection stage. So this is when HIV first replicates into your body after the infection, and in this stage, some people develop flu-like symptoms because there are certain antibodies that are trying to be created to fight off this virus. So that's the first stage, and the second stage would be an asymptomatic stage. So this virus is continuing to weaken your immune system, and the person with this virus at this point will not experience any symptoms, and the average person is asymptomatic for about 10 years. So that's the asymptomatic stage. The third stage is the symptomatic stage, and the immune system is weakened to the point where there is an actual effect where you have symptoms. Person may experience fatigue, weight loss, mouth ulcers, thrush, or severe diarrhea. And these symptoms are normally caused by things called opportunist infections, which basically take advantage of a person's weak immune system. And the last stage, the most severe stage, is something called AIDS / acquired immune deficiency syndrome. So in order to be classified in this stage, a person's CD4 count, cell count must be below 200 cells per mm cubed. And to give a sense of comparison, the healthy adult human has about from 500 to 1,600 CD4 cells per mm cubed. So also, while I'm still on this slide, I want to discuss some of the ways HIV can be transmitted, just to get clear on this. So to start off, basically HIV can be transmitted through any fluid, any type of actually bodily fluid coming from inside of the body. So some examples would be sex, only through that can exchange bodily fluids. Sometimes through someone who's HIV positive sharing certain medical equipment with someone, blood transfusions between HIV positive. I think at this point they actually do test for HIV when you are getting a blood transfusion, just to be safe. And in certain rare cases, the HIV can actually be transmitted through pregnancy from a mother to the baby. So this can be when the baby's still in the womb, through breastfeeding, or through breastfeeding or some other parts. Just at any time when you're transmitting certain bodily fluids and exchanging bodily fluids. So those are some examples of that. So now that I gave a bit of an intro on what HIV is and the stages and a little bit about it, I'm going to go over the structure of an HIV virus in itself. So I'm going to go through the three different types of the structure of HIV. So there's the accessory proteins, the viral enzymes, and the structure of proteins. So first I'm going to go through the accessory proteins. So as you might have guessed, accessory proteins are sort of like an accessory. The first accessory protein is known as the VPU protein, and that stands for the viral protein U. And the viral protein U, to show where it is it'll be around this part of the virus, or of the structure of the virus. It could be it'll be around here. Actually, it isn't shown on this graph, I don't think. But for the next accessory protein, so what the viral protein U does is it basically weakens the interaction between the envelope proteins, these things, when they're attaching to a cell, and it weakens interactions with the cell receptors of the CD4 cell, and it'll also form an ion channel in the viral membrane. So the next accessory protein I'm going to talk about is known as the viral infectivity factor. So that's the VIF, and this is actually, it's written on here. It'll be around here. It's going to be around here in the virus, and so that's the VIF. The next thing we're talking about is the VPR, so known as the viral protein R. So the VPR, basically what it does is it's going to guide the viral genome, so the RNA in this case, into the nucleus of the CD4 cell. So that's important, the VPR. Then we also have NEF, and what NEF is the negative regulatory factor, and it basically what it does is it's going to keep the infected cell from making any self-defense proteins against the virus. There's also another accessory protein that's also on this part written here of this thing, is P7. We actually don't have much information on exactly what P7 looks like or its structure, but what we do know is that it basically it's going to help incorporate the VPR, the viral protein R, into new viruses. And so that's P7. Then we also have REV, which stands for regulator of the virus. And what REV does is it's going to, I don't think it's written on here, but it's going to regulate the splicing and transporting of our RNA. So this is going to exist. Actually, it's going to be around here, somewhere in this part of the structure of the virus. And then the very last accessory protein we're going to be talking about is the transactivator of transcription, also known as TAT. and this is going to enhance the amount of viral protein that is going to be made. And the TAT is going to also be in this part of the structure of the virus, and I also do not think it's written on here. So those are the accessory proteins for the virus, and they all play certain essential roles in infecting of the CD4 cells and the replication of the virus. So now that I talked about the accessory proteins of the structure of this virus, I'm going to move on to the viral enzymes. So these enzymes work in certain processes. So the first enzyme I'm going to talk about is known as reverse transcriptase. So we have this on here, and you can look over here. So what reverse transcriptase does is it basically it builds a DNA copy of the RNA genome of the virus, and it uses it to create more viruses. So in order for the virus to get inside of the cell, the CD4 cell's nucleus, it needs to transform into DNA and be able to integrate as DNA. So that's what reverse transcriptase does. So then the next viral enzyme I'm going to talk about is it's called integrase. And what integrase does, you can see it here. What integrase does is it's going to take this DNA copy of the viral genome and it's going to insert it into the cell's genome, to the cell's DNA. It's going to insert this viral DNA and insert it into the cell's DNA. Integration. So yeah. And so the last viral enzyme of HIV is called protease, and what protease does. Yes, it is right here. And what protease does, it's going to help configure the polyproteins that exist in the virus. So polyproteins are what make up this virus, and they're pretty complicated. And protease sort of is what configures them, and it also plays an important role in the making of more viruses from the cell that it infects and activating them and making them infectious. So those are the viruses, enzymes, and their functions. The last part of the structure of HIV is going to be the structural proteins. So the first protein, the structural protein here is going to be the matrix protein. So it's actually written on this picture here. And what the matrix protein does is it forms a sort of coat on the inner surface of the cell membrane of the virus, of the viral membrane, and it's pretty important when new viruses first show up on infected cells. Pretty crucial. So you can sort of see, on here it looks like little blue circles. So the second structural protein is going to be the capsid protein. That's also viewed here. So it's sort of going to it's the coat that basically it surrounds the virus's RNA, and it's responsible for actually bringing this viral RNA into an infected cell. So what I want to point out is, do not mistake this for a nucleus. This is not a nucleus. It is a structural protein, because viruses do not have nucleuses. This is just a structural protein to protect the RNA. So the next structural proteins I'm going to be talking about are actually very crucial to the beginning of the process of HIV infecting the CD4 cells. So these are going to be the docking proteins and the transmembrane proteins. So they're known as glycoproteins, and they are basically these proteins that surround the HIV structure. And what they do is their purpose is to bind to the receptors on the surface of the CD4 cells. So they're going to bind to the receptors in the CD4 cells, and they're going to penetrate the actual surface of the cell, and then so it can inject this RNA to go inside of the cell. So they're pretty important. One of them docking and transmembrane, and they're going to bind to the receptors on the outside. So you can see them here. So the last structural protein of HIV is the nucleocapsid protein, and you can see this here also. So what the nucleocapsid protein does is that actually it forms a stable structure that actually protects the RNA of HIV, and it actually attaches to some of the RNA. So you can see spiral RNA genome, and then you can see the nucleocapsid. So that is the structure of HIV. So now that we have a sense of the structure of HIV, I'm going to go into a bit about how HIV is going to infect the cell. So the first step for HIV infecting the CD4 cell is the binding to the cell. So in this binding, what we talked about before, these two glycoproteins, the GP120 and GP41, are going to bind to these CD4 receptors. Just to give some information, these little things below this are certain treatments that exist for each of the steps that I'm going to talk about in the next slide. This is attaching to the CD4 cell receptor. So that's the first step, which is binding. The second step is known as fusion, which basically what happens is now that they're connected to the cell receptors, they're the envelope proteins. So these are basically going to attach to the receptors on the CD4 cell. After they had attached to the receptors on the CD4 cell, they're going to sort of fuse together. So you can see this is fusing together, and after it's done this, it's going to allow certain of the HIV content to enter the CD4 cell. So that's the second step, and the third step is known as reverse transcription. And in reverse transcription, the enzyme we talked about before, reverse transcriptase, is used to convert the RNA genetic material of HIV into a DNA genetic material. And the purpose of this is so that the genetic material can actually enter the CD4 cell nucleus, so you can see here, and incorporate itself into the CD4 cell genetic material. So it needs to be DNA to do that, so it's reverse transcriptase. So you have our HIV RNA, reverse transcriptase, HIV DNA, and there are some proteins that come along and help out and keep them stable that we talked about before. So the next step in this virus infection of the CD4 cell is integration. So for integration, what happens is the enzyme known as integrase that we talked about before, what it does is that it takes the viral DNA, the HIV DNA that was created, and it's going to integrate it into the CD4 cell DNA. So we see this here. It's integrated, so integrase into integration. I think that sort of makes sense, the word in itself, as I said before. So that's step 4, the integration. So after that step is the replication, step 5. So in replication, basically HIV sort of uses or takes advantage of the CD4 cell to undergo protein synthesis. So it's almost as if going back to RNA from the DNA. It's going to undergo protein synthesis, RNA protein synthesis, and it's going to create proteins that are sort of a combination of the CD4, like healthy proteins, and the viral proteins. So it's going to create a lot of these proteins, and so these proteins are eventually going to be used to create more of these more of this HIV virus. So that's step 5, and step 6 is assembly. So in this step, these new HIV proteins are going to be used to create sort of an immature version of the HIV. So it's at this point, it's actually not going to be infectious. It's going to be an immature version of the HIV made from these polypeptides that were created in this protein synthesis. So, and the last step is budding. So, in budding, the immature version, you could say, of HIV comes out of the cell, and enzyme known as protease actually sort of activates it to become infectious. And then it's going to keep going, and it's and this virus that has been created is going to go and infect more cells. And at this point, this cell will, you could say in simple terms, explode. And it's going to keep infecting and doing the same process over and over and reproduce. So that's sort of an overview of how HIV will infect the CD4 cells. So now that I've gone through all about HIV and its infecting and its structure, I'm going to go through what treatments, existing solutions that have come through to save people's lives. What solutions are there, and what are their impacts, and what are their limitations? So the main solution that has been developed over the years since HIV has become a problem is known as antiretroviral therapy, ART. So in ART, you're going to take a combination or like a cocktail of different HIV preventing drugs, and you're going to take these normally every day. So as we saw before in the last slide, there you can see these drugs in each of the different processes, and they are exist in each of these different processes. So there are sort of 7 different categories, and most of them, almost all of them, work in separate different processes of when HIV is infecting the cell. So the first one is a nucleoside reverse transcriptase inhibitor, non-nucleoside reverse transcriptase inhibitor, protease inhibitor, fusion inhibitor, CCR5 antagonist, post-attachment inhibitors, and integrase strand transfer inhibitors. So we did explain some of these processes, and you can make inferences into the different parts of the process that these drugs will try to stop. So you might wonder why there are all these different types of drugs. Like, why not just have one drug that will work for all of them? So as I explained before, RNA viruses tend to mutate a lot. So especially for RNA viruses, they can very quickly develop resistance to a drug. So that is why, so normally patients who are HIV positive develop resistance for at least one of these 7 types of drugs. So with all of these different types of drugs, it's much less likely that you might develop resistance to all of the drugs, although there are some cases in which you would. And this antiretroviral therapy has changed everything in the research on HIV. So ART has turned death sentence into a chronic but treatable illness. So to explain this in some actual statistics, in 6 months, certain treated HIV patients are 10% more likely to be active in the world compared to other untreated patients. And now ARV can actually be successfully given to HIV patients actually at any stage of the virus infection, although later stages might have less likelihood of affecting, of having a positive effect, and less likeliness of working out. So yes, these medicines have changed everything, but there are limitations to these medicines. For example, what I talked about before, there is resistance that the virus can that the virus can develop resistance to some or all of these drugs. And with that, without any of those drugs to use, ART would be useless. And so there's that. And as well, a lot of people don't tend to begin ART until much later, until sometimes it's less effective because they were asymptomatic for a very long time and already many of their CD4 cells had been detonated, so their immune system will still be greatly affected. And then there's also the fact that a lot of medicines have different side effects that could actually affect you and give adverse reactions, although the antiretroviral drugs actually have a very, very, very low amount of adverse side effects. So we do have these negatives, we do have these limitations, but I'm not, but these limitations by all means do not stop from saving millions of lives. So now I'm going to get into some of my ideas and some of my opinions on this topic. So obviously, the solutions that I'm going to be presenting are untested and would cost a great amount of money. So I do want to say that they are not by all means in any way proven to work. So first thing I'm going to talk about, so when I first was trying to find a solution to some of these difficulties, the first thing I was trying to look at is how to keep for this virus from mutating, so that these antiretroviral drugs will not have any resistance that will be developed. So the first thing I thought of was, okay, why aren't we just going to add what the DNA polymerase enzyme has, the proofreader, and why not just bring it into RNA and keep it from mutating? But I looked into that in more detail, and I found out that although that would be possible, it wouldn't work to actually be brought into the virus itself. You can't synthetically treat a virus and add that effect, but it's not going to be able to be integrated into a virus itself that's been spreading everywhere. However, this can be used for vaccinations against HIV, and in actual different vaccinations, they actually have, in some cases, they have given this proofreading aspect, adding them to a vaccine for a virus, and it has helped. But in a lot of viruses, this technique has not been used due to actually some adverse effects that can go on. So when you're actually putting some type of inhibitor of a small molecule into a cell that keeps something from mutating, there can be a lot of effects that are very dangerous. For example, there can be lack of platelets, so it's going to keep you from your blood clotting. There can be even a lack of white blood cells, which also would be a problem, especially for HIV patients who already have weak immune systems. So that was almost an option that I looked away from. But then I went into some of the research of the treatments for these specific the adverse effects, and how these adverse effects, when not coming from small molecule inhibitors themselves, are treated. So one of them that's treated for an adverse effect is something called a corticosteroid. Forgive me if I pronounced it wrong. So what this does, there's actually been some research done that from the beginning, certain HIV patients actually responded well to that drug, and especially if it was in combination of another drug. There's also another thing I thought of that actually is there's actually been a research published, a paper on it. I did some research if it had existed. There has been a published, they're basically budding inhibitors. They keep from the budding to these viruses. So it's a new antiretroviral drug that I don't think actually has been FDA approved yet, but it's something that I was thinking about and saw that actually has been starting to be developed. So those are also budding inhibitors. And then I also just wanted to say that I think the most important thing is to stay educated, because the earlier you get your treatment, the more likely it is that you won't be affected. So what I'm trying to do here, the main thing I'm trying to do is to educate people. I want people to understand that that there are many people who might have this virus and have no idea what's going on inside of them. So I'm trying my best, because I think the most effective way to fix this is for education, people to understand and react themselves. So now I'm going to get into my true belief and solution on medicine innovation itself. So I'm going to talk about humanity. You walk around in your everyday life, and you probably aren't constantly thinking about the research and development going on around you. You're going to live, and you're going to take things. You're going to take advantage of things that you normally you don't ever imagine that this has actually been created in a certain way and developed by researchers. So if you do look at certain data, the more innovation we've had, there's been a lot of raising impact in our health. So somehow we've created all this new innovation in medicine, but then to some extent it's actually causing more disease. So to give an example, although this is unrelated, obesity, the obesity numbers have been raising over time more and more as the time goes, and this is mostly due to the fact of a lack of exercise, and people are greatly focused on these new technologies and innovation. And then we also have the fact that many of these medicines that are being created are almost causing more problems. So the solution that I thought of, you can have drugs upon drugs upon drugs that you just need to keep treating, and more of them for the side effects that then have to add to more and more drugs. And the more we create, the more we innovate, the more possibilities of a risk. There have been many viruses, many infectious diseases that have come up that have been brought into humanity because of our innovation, because of our carelessness. So we need to understand that we need to start listening more to nature. And I think the best thing we can do is to stop taking advantage of the things we do every day, and to be aware of everything that's going on and keeping our own opinions and knowing the truth of nature, and that when you have your opinions, that's the most effective treatment. The most effective treatment is to be aware and to act on your own. Thank you.
Interview from the virus point of view.
There's a new treatment. What do I do? What do I do? Where should I go? I can't go anywhere. What should I do? What should I do? What should I do? Where do I go? There's a new treatment. What do I do?
In your mind, do you see yourself as innocent?
I see myself as innocent. It is the blood cells killing themselves. It is not me that is doing it. I just need to reproduce.
Yes. That's all you're trying to do.
Reproduce.
Now, let's take a closer look.
Reproduce.
What are some of the conversations that your outer layers are having?
Talking. What should we do? Should we reproduce?
And why do you like to hide yourself? Why don't you like to show yourself till later?
I would like to have it too late. I would like to reproduce as much as I can until they find me. And I want to have time to reproduce.
Earth. That's where we live, of course. Well, anyway, welcome to the human, interview for the human. And the first question I'm going to ask you is, what do you think of HIV that has been crippling you your entire life? Well, not your entire life since you got diagnosed.
I'm not happy about it.
Why? Why is that?
Because it makes me sick. And they didn't catch it until stage 4.
Do you know how? How do you think people can catch it in time, earlier?
They should be tested earlier. Even if they don't have symptoms.
What about to keep from transmission? What should you do?
Don't exchange bodily fluids with anybody.
At all?
Well, if you do, you should do it in a protected way.
Yes. Now, if you wouldn't mind, I want to take a look at some of your cells that still haven't been infected, and I'm gonna give them a little interview. So.
Okay. Bye.
No, but so I'll talk to you later. Here, I'm gonna talk to your cells. So now, all the way down, and here is an uninfected cell. Hello, cell. Hello, CD4.
Hi, I'm CD4. How you doing?
Hi.
Hi.
And what do you like to do?
I like to do things that CD4 cells do.
Great. Is there something wrong?
No, everything's great. Everything's groovy.
Right? You just performing your processes?
Doing CD4 stuff.
Mm-hmm.
You know, CD4 business.
Yep.
Yep.
All right.
Just going along, CD4.
What if I told you that there's something trying to take advantage of you?
I feel fine. I don't think there's any problems here.
All right.
Everything looks good.
Okay.
We're good. Well, it's all good, man.
Yep.
Yeah.
Nice. And how do your receptors feel right now?
Never better. Never better.
Why is that?
Because they're clean and free and working great. They're receiving.
What do you think of viruses? Do you think they're bad?
I never heard of viruses. I've never seen one. I
Well,
don't even know what a virus is practically. I'm CD4. I just do CD4 things.
yeah. All right.
1, 2, 3, 4.
Well, signing out the interview now. Signing out. Goodbye.
Bye.
Has invaded you and has integrated into you. What do you think of it?
I used to be a happy and functioning cell until that horrible virus. And now, all of my functioning is only a faint memory, and I'm slowly falling apart!
Well then, I was going to ask another question, but the cell has died. And yes, the cell shouldn't normally know that the cell itself has died, but this cell is a smart cell. Not anymore, obviously. Well, that concludes our infected cell interview. What do you think of these HIV viruses?
Oh, I'm gonna get those viruses. I'm gonna inhibit their reverse transcriptase.
Oh yeah, you're a reverse transcriptase inhibitor?
I'm gonna do that. That's what I am.
Yeah.
I'm gonna inhibit the heck out of them.
Yep.
I'm gonna inhibit them here. I'm gonna inhibit them there. And even if they mutate and they become resistant to me, I have like 6 other friends who will do their own things. There's no way these viruses.
You don't think the virus is just innocent?
There's no such thing as an innocent virus.
All right. Anything else to say?
Just HIV, you better watch out. I'm not even going to tell you what I'm going to do to your reverse transcriptase.
Okay. Thank you.